I am very happy to welcome my good friend Andreas Nieder, played an instrumental role in the discovery, simultaneously actually with other Japanese colleagues, but with Earl Miller at MIT, the discovery of neurons that care about number. And so today he's going to tell us about the neural coding of number and other dimensions.
Yes, so thank you, Stan, for the introduction. It's a great pleasure for me to be here at the Collège de France and to celebrate with Stan and my colleagues 20 years of the snark effect. After Giorgio's brilliant talk, I hope you're all convinced that also animals can discriminate numerosities and have some form of numerical competence. That's great because I need this for my talk as well. I hope I can contribute two new aspects. One is not a human primate as a model system, but the non-human primate, rhesus monkeys. And the other is a different level of explanation, namely the level of single neurons that I'd like to present. What are our research questions? I try to highlight them here. So first of all, we would like to find out where and how in the brain numerical information, particular quantity information, spatial information is represented. And secondly, how abstractly this is represented, because after all, the concept of number or numerosity should account for all modalities across space and temporal presentation. Then we wonder how numerical, spatial, relational quantities relate to one another. Is it possible to find neurons that are encoding different aspects of quantities, different sorts of magnitudes? What happens when numbers are related to signs? That's something I will not go into deep detail today. And finally, that's our sort of latest research direction. We know that numbers are great, numbers rule, but numbers alone, just representing numerosities, is not enough. They have to be processed according to rules, ideally, to allow rule-guided behavior in executive functions. And that's what I'd like to address in the end of the talk. Most of the data I will present, most of the presentations of numerosities I will present are from the visual domain, so this is just a brief reminder of the visual system that we are dealing with. This is a human brain, one of the few human brains you will see throughout of the talk. Of course, visual information enters the brain via the eye, then we have visual cortex at the back pole of the brain. But this is not the end of the visual processing, that's just the beginning. Here at the bottom you see a rhesus monkey brain with the ventral pathway leading towards the temporal lobe, which is thought to convey mainly identity information, identities of visual objects. And then the other path which will be more important for us, the dorsal pathway, which is thought to represent spatial information and visual information used for action. Both of these visual pathways converge on the prefrontal cortex, which is our and other primates' highest cognitive center in the brain, the executive master, if you like, that guides our behavior. And I will show you data from both the prefrontal cortex, where we recorded from single neurons, and the fundus of the intraparietal sulcus, deep in the intraparietal sulcus, mainly this area, VIP, ventral intraparietal area, because as it turns out, there are particularly many numerosity selective neurons. As I said, we are interested in the level of explanation of single neurons. Single neurons convey and process information based on minute electrical discharges that run along their axons. And this this animation nicely illustrates the complexity with which we have to deal. If you scale this up and have it about 10 times faster, then you get a rough idea of what's happening in the cortex. All these little light flashes here would be electrical discharges that we try to detect with our microelectrodes in this mesh of axons. So we use microelectrodes, they are as thin as a hair. We bring them close to single neurons. The electrodes can pick up the electrical discharges, and we stimulate while monkeys are working, usually visual stimulation, at the same time record from these two areas in the brain the electrical discharges, and we may detect a certain number of action potentials for one representation of a numerosity and yet another number of action potentials for another numerosity. And in fact, we count the numbers of action potentials over time and derive the rate of action potentials and use this as a measure of how well neurons represent information. The standard task that we use now almost, well, not almost, but more than 10 years is a delayed match to numerosity task, or a delayed match to numerosity task. So the monkeys are sitting in a darkened booth, they are facing a computer monitor and perform this sort of computer game. They fixate to start a trial and grab a manipulandum. After that, a sample stimulus appears, cueing the animal for a certain number of items, 4 in this case. Then they have to remember it over a brief delay period, so working memory is activated. And then comes a test stimulus, which in 50% of the cases is a match. It shows the same numerosity, but importantly with a different visual layout. So the monkeys can't use the perceptual layout as a cue to discriminate. And here the monkeys is required to release a lever to get some reward. In the other 50% of the cases, a non-match is presented showing a different numerosity, and here the monkey learned to maintain the manipulandum and wait until the second test appears, which is always a match. So according to this protocol, we have a chance probability of 50%. Everything better, performance better than 50% would indicate that the animal can discriminate numerosities. When you look at these images, multi-dot images that we have seen in the morning already, it's evident that there are many non-numerical, purely visual parameters that may co-vary with an increase of the number of dots in these displays, and we have to control for that. Of course, here are all the controls that we used over the years, like controlling for the total area of the black dots, the circumference, the density, and so on and so forth. I won't go into detail, but just to indicate to you that we are aware of this problem and we do everything we can to exclude these factors, and I can assure you that neither the monkeys nor most of the neurons in the end care about these purely visual aspects. They really do care about the numerosity. What we find then indeed in these two areas, prefrontal cortex and ventral intraparietal area in the intraparietal sulcus, are neurons that are specifically tuned to one of the numerosities that we present to the monkey. And here is one example, and the layout will be shown over and over again throughout of my talk, so I just guide you through. What we see here is the discharge rate of a single neuron plotted against the time of a trial. At 500 milliseconds, the sample stimulus is shown, and then here the delay period starts. We present, in this case, 5 numerosities several times to get rid of the neuronal variants. And you see color-coded here, every line here represents the discharge of a neuron. Every dot is an action potential. And you see that these are color-coded for the specific numerosities. The same type of information is averaged and summed here in this spike density histogram. And I hope you can appreciate that this neuron hardly reacts to numerosity 1, even more better to numerosity 2. 3, pretty high, that's the green one, then the black one, that's 4, that's the highest discharge, and for 5, the purple one, it drops off again. When we integrate over this period, we get what is called a tuning function. Discharge rate is plotted against the number of items, and this, in this case, this neuron has a preferred numerosity of 4. It discharges the most to 4, and for smaller and higher numerosities, it drops off again. We tested a broad range of numerosities so far, up to 30, numerosity 30, and for all of these numerosities that the monkeys are faced with, we find specific neurons that are tuned to one of these numerosities, and here are 8 examples. That's a neuron that's tuned to 1, 2, 4, 6, 20, 20, 26, and 30. So this tuning behavior, that's something we encounter over and over again when we record together with the group from Japan, Yun Tanji's group, with a very different task. They also find this sort of Gaussian type tuning functions. And this is the moment where I have to disagree with David Burr as much as I admire his work. But both in the behavior and the neuronal representations argue in our hands for a labeled line code that is non-linearly compressed. And the reason why I tend to think this is that both in the behavior and in the neuronal data, a logarithmic compression of the number line, if you like, is better describing the data. So on the left-hand side, you see the behavior. All these functions indicate the probability a monkey detects a certain target numerosity from 1 to 6, or a certain numerosity as being equal to this target numerosity on the top here, giving rise to these Gaussian type performance distributions. And you may see that they are not completely symmetrical when plotted on a linear scale, but they become nicely symmetrical when we switch the scale to a log scale. And we can also quantify this. We see the very same effect for the neurons. Here we averaged and normalized all the numerosity tuning functions of the neurons we encountered and plotted them in such a way. Again, we find that a linear code, a linear representation of the tuning functions is worse than a logarithmic function, worse meaning that only in the logarithmic situations the tuning functions become symmetrical Gaussian functions. And that's why we think that both behaviorally and neuronally a logarithmic description, or at least a nonlinearly compressed description of the neurons or we prefer over a linear representation. Where do we find these neurons? We recorded in the meantime from a variety of areas, both in the temporal, parietal, and prefrontal cortex. Here, that's a lateral view of a sketch of a monkey brain. The color here indicates the probability with which we encounter neurons that are tuned to specific numerosities. And you see immediately there's a high percentage of numerosity-tuned neurons in the prefrontal cortex and deep in the fundus of the interparietal sulcus. And I should mention that we collect these neurons in an unbiased way. So we pick every neuron that has a good signal-to-noise ratio and later on see whether it was tuned to numerosity. So this is a quite realistic sketch of what's going on in a monkey's brain. Of course, when we have these two areas, then the big question arises, could we pin down a hierarchy? Is one area earlier representing numerosity than another one? With single-cell recordings, we have the advantage of very precise and fine temporal resolution. So we calculated the time point at which the population of neurons in either of these areas became significantly tuned to numerosity. And it turns out that the neurons in the parietal lobe are way earlier representing numerosity, like 50 milliseconds earlier than the prefrontal cortex. So our current working hypothesis is that this is really the first place in the brain of a primate where this information is extracted, and then it is routed forward to some more high-level executive structures like the prefrontal cortex to be available for goal-directed behavior, working memory, decision-making, and so on and so forth. Another important question, of course, is this in any way related to what's going on in the human brain? We've seen some of these images already. I just would like to highlight one meta-analysis that Stanislas Dehaene did a few years ago, collecting many, many functional imaging data and determining the overlap in all of these different studies on the brain, where in us humans, and that's a slightly inflated version of a human brain, activity is present when we detect numerosity, discriminate numerosity, deal with numbers, solve equations, calculate, and so on and so forth. And I hope you can see that also in us humans, the posterior parietal lobe and parts of the prefrontal premotor cortex are activated. So what we find in the macaque monkey brain is most likely, at least that's what we tend to think, a homologous structure of what later on develops into a much more sophisticated system, of course, in humans, particularly when the language system kicks in. Now, so far, I only told about this, what we call simultaneous protocol, presenting dots of objects at the same time so they can be assessed at one glance. But when you think about counting, real counting, then this is actually a sequential process. We count 1, 2, 3, 4, 5, and so on. So we wondered whether the neurons that we encountered would also detect the numerical information, the numerical quantity, when the items are presented in sequence one after another. So we trained animals, in addition to this protocol, also to detect the number of items from 1 to 4 in this sequential protocol. Here, of course, we have to be very careful not to allow the animal to solve the task based on numerical information, sorry, on temporal information. And again, I just would like to assure you that we did everything to destroy temporal factors that could allow the monkey to solve the task. And indeed, we found neurons in area VIP that particularly after the last item has been shown and the delay period starts, were encoding numerosity irrespective of whether it has been presented in this way or in that way. So neurons that were tuned to the same numerosity, both in the simultaneous and the sequential protocol. One reason also to implement the sequential protocol was because we wanted to move on from a purely visual presentation to also an auditory presentation, because after all, as I said already, 3 pictures and 3 calls are all instances of the same numerosity 3, and if these detectors are really abstract, as we like to think, then they should no longer care about the modality, and they should integrate across modalities in a supramodal way. That's what we did and published recently. So in addition to this protocol, the sequential visual protocol, the monkeys were trained to detect the number of sound pulses, white noise pulses from 1 to 4, and match them after a brief delay period to a visual test display. And indeed, we found neurons in both area VIP and prefrontal cortex that were nicely tuned to visual and auditory numerosities. Here is one neuron and its tuning function. So this neuron doesn't care about whether we show 2, 3, or 4 dots in a row. All it cares about is the first one, and it behaves exactly the same way when we present auditory stimuli. And the second neuron, that's another example, that prefers 3 items in a row or 3 sound pulses in a row. And in both instances, it provides the information 3 objects or 3 events. Of course, not all of the neurons were encoding numerosity in this multimodal way. Here is a sketch, a summary of what we find in area PFC and VIP. So in VIP, about 10% of the neurons were only auditory or only visually representing numerosity, and only a very small overlap of 3%, which was not statistically significant and what you would expect based on these probabilities. So in area VIP, we had a hard time to find these supramodal detectors, but it looks different in prefrontal cortex, which, as I said, is higher up the hierarchical level. There we find more neurons that are unimodally tuned to numerosity, and significantly more neurons than would be expected based by chance that are encoding the same preferred numerosity in the visual and in the auditory domain. So here we would speak of supramodal encoding of numerosity, and these are really very, very abstract encoders that no longer care about the sensory modality of the numerosity. This meeting is also about other sorts of magnitudes, particularly spatial magnitudes, and we tried to address the question, why could numerical information and spatial information be somehow related to one another? Because at first sight, I think there is no reason why space and number should have any commonalities. So what could be going on in these areas? So we recorded in area VIP and PFC from monkeys that were trained both to discriminate numerosity as a sort of discrete numerical magnitude, and at the same time from a different sort of magnitude, a spatial magnitude, and we took the length of a line here. So 4 different lengths and 4 different numerosities, all in the same session, intermingled, and we could confirm that the animals were not associating numerosities with line length. So for them, they were completely different magnitudes. And again, we found neurons. I show you here 3 neurons that were nicely encoding the numerosity in these displays, but completely uninterested in the length stimuli. Yet other neurons were nicely tuned to the length of the lines, like this one was detecting the 3rd longest line very nicely in a very tuned way, but not at all interested in numerical information. And then the most complex type of responses, the 3rd neuron here was encoding both types of information in a very unrelated way. Now, looking at this data, there is one good thing and one bad thing. The good thing is, yes, the neurons in this area are encoding both numerical and spatial magnitudes. The bad thing, however, is that when you look at the responses of single neurons, they cannot disambiguate what magnitude has been shown and what value of the magnitude has been shown. They are ambiguous encoders. So we wondered whether the entire population of neurons that were significantly tuned to these magnitudes could provide us with the information that the animal would need to solve the task. And that was our next step. So here we made a population coding analysis with a statistical classifier, a cohort network. We trained this network with 90% of the spike trains that we recorded from 71 best neurons. And in this training phase, the decoder was allowed to know what numerosity or what magnitude and what value of magnitude has been presented to the animal when the specific spike train was recorded. And after the classifier learned to take as much information as possible out of the spike trains, it was tested with the remaining 10% of the spike trains it had not seen before from the same animals. And the question was, would the classifier, as a sort of an ideal observer now, be able to detect the magnitude and the value of the magnitude? That's what's plotted here on this confusion matrix. On the left-hand side, you see the true class. So these are the displays that have been presented to the animals when a specific discharge was recorded. And here is the predicted class predicted by the classifier. And you see, according to the blueness or to the darkening of these fields here, the prediction made by the classifier based on 72 best neurons. And I hope you can appreciate that most of these hits are really on the diagonal in this confusion matrix, and that's something that we would sort of predict if the classifier would be able to encode or to extract the true magnitude that has been shown to the animal based on the firing rates, then all the data points should lay on the diagonal. Of course, because we only have a relatively restricted number of neurons, it can't be 100%, but it's more than 60% at a chance level of 12.5%. That's highly significant. So yes, based on the entire population of neuron, the monkey could have all the information it needs to solve the task. And we also verified that this information is really behaviorally relevant, because when we fed the classifier after the training stage with the trials where the monkeys made an error, also the classifier dropped dramatically in performance. So here I'd like to switch gears a little bit and address the last topic of my talk. This is now going away from representing magnitudes towards processing magnitudes. Because if you imagine, we and animals, of course, we are not happy with simply representing numerosity, we need to deal with it. Or imagine you're a chimpanzee patrolling at the territory borders of your territory, and you have to decide whether you need to respond aggressively to an intruding group or to retreat to save your life. And at that point, you need to compare the number of intruders to your own party members and decide based on the relative strengths in numbers whether to retreat or to attack. Rules, in a general term, would be conditional relationships that describe the logic of a goal-directed task. Rules in mathematical or quantitative terms can be very easy, like less than, greater than operations. That's something also children learn in first grade. And this is a task that also monkeys just master, so they can do it. This is a task they, of course, can no longer master. But this is just to emphasize that in highly formal mathematical systems, of course, we need to apply and learn rules to know how to process these equations in a goal-directed way. So rules are really important, particularly when dealing with numbers. So we are using this task with our monkeys, and we trained them in a rule-switching task. They had now not, their job was not to match numerosities, but to pick more or fewer numbers of items according to a rule that was presented to them in every single trial. So the monkeys again in the booth grabbing a lever, fixating, then a sample image appears. In this case it's 5. The monkey has to remember the 5 dots. Then the rule cue appears, that's this red circle. This cue means that as soon as you see a second numerosity display, pick the one that is higher in numerosity, and do not respond to the one that is lower in numerosity than what you have seen in the sample numerosity. So the monkey is supposed to respond to this display, not to that one. The alternative rule would be the blue circle. Here the monkey is required to respond to fewer items and to maintain the lever if more items appear. From a behavioral point of view, we would be done here and could record the behavioral performance. From a neuronal point of view, we had to do a little more because we are really interested in what's happening after the rule cue had been displayed to the monkey in this display 2, because this is exactly the phase where the monkey could use and apply this principle it had learned, this abstract rule, but it cannot yet prepare a motor act because it needs the information from the second display to prepare and to decide how to react. So this is a phase where we can really, as purely as possible, study this representation of an abstract rule. But to do so, we have to make sure that what we detect here is not a function of the color, because as you may see, this would be confounded by color, red here and blue here. So in addition, we introduced a second set of rule cue sets from a different modality. A white circle with drops of water indicates the greater than rule, and a white circle without drops of water indicates fewer than, the less than rule. And in that way, we can factor this in our analysis and make sure that we are only detecting rule activity and not some low-level visual activity. And of course, we train the animals not only to respond to 5 numerosities relative to 5, but to a broad range of numerosities to make sure that they understand and grasp this abstract concept of greater than, less than. So we end up with a 4 factorial design. I don't want to take you through all the details, just to indicate that we can control for all these factors. And what we are looking for are now responses that only encode the rule, but no other factor here. And indeed, in the prefrontal cortex, we found such highly abstract responding neurons, like this one here. This is a less than rule selective neuron. This is again a spike density histogram. That's the average activity of a single neuron throughout the trial. And as I said, particularly interesting for us is the delay 2 period, because that's the first time where the monkey has the rule available, but cannot yet prepare a motor act. And you see very nicely the dark colors here, that's the less than rule, presented in the 2 different modalities. It nicely increases in activity, but it doesn't do so for the greater than rule. This is a magnification of the same response period. An example of a less than rule, we also found greater than rules, about as much as less than rules, funny enough. About 10% of the neurons in prefrontal cortex are encoding the less than rule, and the other 10% are encoding the greater than rule. The remaining 80% are involved in other stuff that the monkey has to do, of course. So we end up with these 2 populations of rule selective neurons, and we were wondering whether we can say anything about the evolution, the temporal evolution. When do these neurons become active to guide the monkey's behavior when the reference numerosity appears? So what we did here is to calculate the so-called receiver-operator characteristic, a measure derived from signal detection theory. Without going into the details, ROC value of 1 would indicate that the neuron is perfectly able, based on its firing rate, to discriminate a less than and a greater than rule. By definition, 0.5 would mean that the neuron can't discriminate at all between these 2 rules. And the blue function here, these are all the neurons that have been classified as less than neurons. So blue areas mean neurons that are encoding the less than rule. Red areas are the neurons that are encoding the greater than rule, plotted against the time throughout this second delay after the rule cue had been presented for all the neurons that turned out to be significantly tuned. And you see, the longer this period continues, the more rule selective neurons kick in, probably providing the monkey with more and more confidence about what rule to apply as soon as the comparison stimulus appears. And that's my final slide, final data slide. We not only recorded from the prefrontal cortex, but later on also from the intraparietal sulcus area, VIP. The result was relatively disappointing. Also from a premotor area and the cingulate motor area deep in the middle prefrontal cortex. The result is depicted here. In the frontal lobe, we find significantly more neurons that are encoding this abstract quantitative rule compared to the other areas, including VIP. So here, VIP is probably not very helpful. It has to be transferred to the prefrontal cortex to gain control over behavior. And with this, I'd like to conclude, summarizing. So there are, in fact, neurons both in the interparietal sulcus and in the prefrontal cortex, and maybe even at other places that we haven't yet explored, that encode numerosity and other quantities in a very abstract way, abstract here meaning across time, space, and modality. We find a distributed but overlapping coding of different types of information, meaning that they are represented by neurons in relatively restrained neuronal areas in area VIP, but immediately adjacent neurons can have very different preferences for magnitudes. But still, because they are functionally and anatomically heavily intermingled, maybe this provides a reason why there is this interaction between numerosity and space. At least this would be one explanation. Neurons, I didn't talk about the third conclusion, so I skip it. The frontal lobe neurons can nicely and flexibly encode quantitative rules that I've showed you over the last talk, part of the talk, and all that together argues that what we find in a non-human primate is, at least on a non-verbal level, quite reminiscent, probably homologous to what's going on in our brain before the symbolic language system kicks in and raises our number understanding to a completely different level, of course. So thank you for your attention.